Are we? Okay. Hi, everyone. I am Kristen Baum, and this is Casey. And we are the founders of the nonprofit Clara Inspired, but more importantly, we are Clara's parents. Clara's our STXer. We were asked to share our story with you today, from diagnosis to dealing with grief to running a million miles, not really, but kind of, raising a bunch of money, and inspiring as many people as possible along the way. So Clara was born on December 3rd, 2013. Her birth was exciting, but pretty uneventful. She was born on her due date. And we were thrilled to add a second daughter to our family. Clara's sister Avery is 3 years older. She's now 14. She's not here today because teenagers. She's usually here though. So Clara's first year was pretty typical. Honestly, we didn't really have any concerns except for, a couple of developmental things. She lagged a little bit behind her sister and her sister's development, but, not any huge concerns. And as more time went on, more milestones were unmet, and we started looking for answers. So we met with a developmental pediatrician who eventually sent us to a geneticist. I'm sure this sounds very familiar. And we heard a lot of she'll catch up eventually and nothing to worry about. But eventually our doctor decided to send us to a geneticist to test just in case. So fast forward to April 2016, Clara was 2.5, and we finally received the results of our whole exome sequence. Clara had, you guessed it, a mutation or change on her STXBP1 gene. In 2016, there was only about, 100 to 150 of us, so definitely a small group. So the doctors sent us home with a Google printout, and we started the next phase of our lives, which was post-diagnosis. Slide 3. So I immediately went into crisis mode. We did a benchmark EEG right away. At the time, Clara was not having seizures. Unfortunately, she is now. We also did intensive physical therapy sessions. I spent hours online trying to find a sliver of information, and of course, begging to be let into the Facebook group, like literally begging. I asked to be joined, and then I messaged people right away. And that's where we finally found our people. It was a small group at the time, but very mighty, and some of these people have become our family. Casey, on the other hand, also went into crisis mode, but channeled it in another direction. He looked at our daughter in the midst of grieving, the life we thought we had, and he felt inspired. Clara was his inspiration, and if she couldn't grow up to have a typical life and a typical profession, do all the typical things, then we were gonna help her get her first job, Chief Inspiration Officer.
Yeah, so in the 2017, 2018 timeframe, I didn't quite know what to do or how to handle this, and I wasn't much of a runner. I was kind of an athlete in high school, but hadn't done anything in 15 years, just kind of hung out. But this was kind of the perfect opportunity to sort of get back out there and kind of start spreading the word a little bit. So, Clara was recently diagnosed, and we thought we'd get the word out a little bit. So I had a friend who was running up in Boston, and marathons to me seemed like no fun whatsoever. Why would anyone want to do that? But he sort of talked me into it, and I was like, well, this is a good opportunity to raise awareness for STXBP1 and the new diagnosis that we have for Clara. So we ran a marathon in 2017 with our STX shirts, and everyone thought it was great. But they were like, yeah, that's great. I have a friend who ran a marathon. I have a family member who ran a marathon. I was like, okay, but I'm running the marathon because it's hard, but we're doing it because raising a child with special needs is really difficult, as all of you here know. So I'm like, this is really hard. And they're like, yeah, but marathons, they're okay. And I was like, I don't think the message is quite getting across that it's hard to raise a child with special needs. So I was like, well, what's harder than a marathon? So I started kind of doing a little research and found just a few hours from here, there's a race called the Leadville 100. It's 100 miles. And you have to kind of enter a lottery to get into it and get selected. The chances of getting selected are very small. It's 5% chance, 10% chance that you get picked for a lottery. So this is my first time. I don't know any better. I think, well, let's go ahead. I'll just throw my name in the hat. Like, I ran a marathon. I'm sure I could do something else. Sure enough, they select me to run 100 miles. I was like, okay, well, this is something new for me here. So what we did is in the 2018 timeframe, we formed Clara Inspired. So Clara Inspired was born in 2018. And the message that I wanted to get across was that, we can do hard things. We see all the hard work that Clara does through therapy, and we see all the hard work and all the difficulties that everyone in this room has experienced. So we would talk to each other, and we'd kind of have a little internal motto that was like, well, today was difficult, but we can do this. Like, today was hard, but we can do hard things. So I wanted to really sort of prove that we could sort of put ourselves out there and do a really hard thing. So sure enough, in 2019, went out there, didn't really know what I was doing, but 29 and a half hours later, we're able to finish, go across the finish line and raise about $50,000 for the newly formed Clara Inspired. Thank you. And sort of get the Clara Inspired story going. So, from there, we've sort of continued throughout the years. Kristen kind of took the reins after 2019, and she ran.
Like, I can run too.
She ran a marathon during COVID, which was very difficult because it was kind of like a virtual. You kind of do it on your own. She just crushed it, and then we've had friends and family along the way who have helped us. So in 2022, Clara ran her first marathon. This was a team of six of us. Again, raised some money and pushed Clara. It was we're in Richmond, Virginia, and this was November. So you're thinking a 40-degree day, like this should be fine. And sure enough, it was like 81 degrees in November. We're like dumping water on our heads throughout. It was almost, no kidding, it was almost more difficult than the Leadville 100. Yeah, and since then,
we have done a lot of running, and we're getting tired. So if anybody has any suggestions on other things we can do, please let us know. But we have done a few other things. After Leadville, in 2020, we obviously, I think everybody was inside, and so we were trying to figure out what our next fundraiser was. And Casey, who comes up with the crazy ideas, and I always say they're crazy right away, but then I'm happy afterwards, he also plays the guitar. So he decided to do an 8-hour guitar session. So he played online for everybody for 8 hours, and we raised like $20,000, I think, that day, which was cool. So we've done that. We've done running. Clara's involved. So just a lot of fun things. And I think the most exciting part is watching our friends and family also get inspired and start doing things that they never thought they could. It doesn't have to be a marathon. People who haven't ever run before, just running, small little races or walking or, just doing things that are hard. And so it's been really exciting. We've also have a merch store, so if you want Clara Inspired gear, feel free to get that too.
Yeah, and I'll just say that none of this would be possible without Clara. None of this would have happened. So, she is our inspiration. She's the reason why we're doing this. So. We want to thank her for being the Chief Inspiration Officer, and this is what her profession will be for the next several years. And we're yeah. But sort of to wrap up here, the money that we've raised over the years have gone to various causes. We've worked closely with Charlene and the STX Foundation to contribute to some of the clinical trials, and our thing a year or two ago was we bought a mouse. So that's bullet point 3 here, which we thought was pretty cool. And we know that there's a lot of people who do so many things for this organization, this community. We're happy and just thrilled to do a small part from there's so many other things that everyone is doing. So. We're just so grateful and thankful to contribute as we can.
Yeah, you said it.
Thank you so much. Appreciate it.
Thank you.
All right, thank you, Baum family. So you guys heard a lot about our disease concept model, and one of the key concerns that was identified in our disease concept model is behavior. And so we have another representative of the Children's Hospital of Colorado, Morgan Jolliffe, who is going to give us a little presentation on behavior in STXBP1. So.
Hello, I'm Morgan Jolliffe. I'm a neuropsychology fellow at Children's. Thank you so much for that introduction, and looking forward to being here today. I'll be speaking about behavioral symptoms of autism in STXBP1. So wanted to credit my team, my PI and mentors, Andrea Mile, who was able to speak earlier. We're sharing some clinical data from Children's, but also some data from the STAR study. So I wanted to thank my co-investigators at CHOP, especially Carlene Glatz, who's not here today, but was really instrumental in figuring out with me how to look at this and how to interpret the findings and stuff. So thank you to CHOP, and yeah, I'll jump in. So the purpose of today is just to give everyone an update on where we are with assessing behavioral symptoms of autism in this population, kind of present a little bit of data and talk about what we're learning about how to best use these measures, and talk about what's coming in the future too. All right, so as you know, behavioral features of autism are commonly reported in STXBP1, but formal diagnosis is really complicated by overlapping symptoms like lower social communication skills, and there's a lack of well-validated assessment tools in this specific population. So because of that, we suspect that there is some underdiagnosis, and accurate diagnosis is incredibly important for informing targeted early interventions, opening up doors and access to services, and all of that. So we definitely want to better sort through our most accurate measures and not be missing diagnoses, so that we're not delaying any type of early intervention that could shift the trajectory for social and language development. So before we jump in, I just want to make sure we're all on the same page with what kinds of symptoms and behaviors we're trying to describe. So autism is a behavioral diagnosis, so it's based on symptoms, not based on etiology or why the symptoms are occurring. And as long as there are sort of enough criteria in these two buckets, so social communication interaction and restricted interest and repetitive behaviors, we would classify the combination of those types of symptoms as autism. So to give you some specifics on what those symptoms may look like, social communication weaknesses may include limited or reduced eye contact or facial expressions, joint attention or reduced social smile. There can be delayed or absent nonverbal gestures, or there are gestures maybe poorly integrated with speech. A lack of imitation skills is quite common, reduced language for social purposes, and sometimes the lack of social motivation, although we don't always see that. Within the restricted interest and repetitive behaviors category, we think of things like repetitive motor movements, so spinning or hand flapping, can be head banging, things like that. There could be repetitive speech, which could be like speech for non-functional purposes, kind of repeating things that have been heard. Inflexible patterns of behavior, so rigidities, kind of getting stuck, and also sensory sensitivities. And then those arrows on the bottom just kind of depict that within this diagnosis, there's a wide range of expressive language skills, so ranging from nonverbal all the way to highly verbal with some notable social communication differences, and same with the level of support. There's a wide range, kind of shifting from needing to depend on caregivers all the way to maybe needing little or no support at all. So the purpose of our study, kind of our long-term goal, I guess, is to clarify the presence of autism and make sure that we're not sort of under-capturing it in this population, and help clinicians feel more comfortable diagnosing autism. So to give a little bit more assurance that these are the right tools. In order to do that, we have lots of work to do. So some of these things are what we're working on now. Of course, we're gathering data every month, kind of adding to our study. But we are really trying to describe how individuals with STXBP1 are performing on these measures. We're looking at how these measures correlate to one another. So not only do we trust this measure, but does it align with some of the other pieces of data that we're gathering? And then a really important one is number 4, which is determining the degree to which these measures are picking up on other broad developmental difficulties like lower cognitive skills or language skills rather than autism-specific skills. So we have access to 3 measures at this point: the social responsiveness scale and the BASC, Behavior Assessment System for Children. Those are both parent-reported measures. Parent data is incredibly important to us. We know that we only see them in a brief assessment setting, which doesn't really mimic the real world and kind of the experiences that parents are seeing on a day-to-day basis. So those first 2 are really extra important to us. And the CARS 2, this is a clinician-rated measure, and this is filled out by a clinician who's attuned to paying attention to the specific behaviors that we're talking about. They integrate information from parents, from their own observations to fill out the CARS. And this gives you an idea of kind of our sample size. So in the larger STAR study, we have, you heard earlier today, over 100, but with these behavioral measures, we have a slightly smaller N that we're working with, but enough data at this point to do some analysis. So first, the social responsiveness scale. Here I've plotted out the means for each of these indices. And just to orient you, the T-scores indicate more impairment, so a higher T-score would be more symptoms of autism. And the blue line is sort of where our means are falling. The colorful green, orange, and red are kind of indicating different cut points. So anyone below 60 would be scoring within sort of normal limits. And then between green and orange would be like mild symptoms of autism, 65 to 75 would be like moderate severity, and then 75 and above is the most sort of severe. So on average, for most of these subscales, so we're talking about social awareness, which is what it sounds like. Social cognition is like the ability to think about what someone else's perspective may be. Social communication, also what it sounds like, and repetitive interests and behaviors. All of those are kind of approaching the severe range. And then, of course, all the indices. So SCI total would be all the indices, the subscales that relate to social communication, and the RRB total is a larger index scale as well, and the SRS total includes everything. So as you can see, we're reporting a lot of different symptoms. The one that you'll see that dips down and that's very preserved is social motivation. So we're seeing that fall in the normal range. So within STXBP1, social motivation is largely preserved, maybe a strength that we saw this earlier at AES. I presented on the SIM topic with a lot less people. We're seeing the same pattern, so this is just kind of an update to that. And then we also looked at the SRS compared to the Bailey to see if we're picking up on broad developmental problems or autism specific, and we had no significant correlation with the Bailey, so that was reassuring for the SRS that we are indeed looking at more of those autism specific behaviors and not broad delays. On the BASC, there's a number of clinical scales, but the ones that are noted to be often elevated in autism are the atypicality, which is sort of a broad, it includes less social interaction. It also includes some strange behaviors and repetitive movements. So atypicality you can think of as kind of a broad autism indicator, and we are seeing that fall between orange and red, which is sort of in this at-risk or elevated range. While withdrawal, which is more of a social isolation, that one is under the yellow or the orange line, which means that that one was within normal limits. So if you think about the comparison, also this one was not correlated with the cognitive skills at all, so that's great to see. But when we compared the SRS and the BASC, we are really reassured that we're kind of measuring the same thing, or we're measuring what we think we're measuring. So the SRS total was moderately positively associated with the BASC atypicality score. So not only are both of these measures not sort of implicated with lower cognitive skills, but they're associated with one another. And same with social motivation and withdrawal. So we're more confident too in that suggestion that this is sort of a unique profile of autism where there's not a lot of problems with the withdrawal, or people are more motivated, because those were positive to moderate to strongly associated as well. And then the third measure that I wanted to talk about is this clinician-rated measure, the CARS. So on this one, we have a group of 35 people that we have CARS and Bailey scores for. And this is the piece of our work that we're doing with CHOP actively right now. So each bar represents an individual, and we have raw scores plotted. So anyone under 30, sort of within this normal limits, we only have 2 individuals that were falling into that group. Most people assessed are falling above 30, so between 30 and 36 is sort of mild to moderate severity symptoms of autism, and above 36 was in that more severe range. So 50% fell above 36, so a lot of people are falling into that range, and 94% are at least mild to moderate symptoms of autism. So similar to what you saw in the BASC and SRS, we're capturing lots of these symptoms, but our next question again is this one related to cognition? So first, we looked at individuals that had taken this measure that were over 2 years of age, because it is recommended that this measure is used for kids above that age, so that for that same reason, so that we're not picking up on areas of development that haven't developed yet. So we did see a significant relationship for these kiddos between CARS and cognitive ability, and between CARS and receptive and expressive language. So we then decided, because we do see some risk for overdiagnosis in this range and misclassification, we decided to look at individuals above 30 months, so that's 2.5, give a little bit more of a buffer. And when we looked at this group, those effects disappeared. So above 2.5, we're not seeing a relationship between autism symptoms and cognitive functioning. Although we are still seeing a relationship between cognitive functioning and language development. And that is expected because language weaknesses are a core aspect of autism. So this makes a lot of sense and gives us some assurance that these measures are working well, especially when we are careful not to use them in too young of a population. We're still a little wary. These are very preliminary findings because the individuals that fell within that 6-month range, it was a small group, only about 5. So it was a little surprising that the effects completely disappeared when we took out that small sample. So we are curious if when we add more people, if these kind of results hold up. The last thing that I wanted to share is some item level analysis. So we kind of dug in and saw what are the most highly rated items on the CARS. And we looked at specifically anyone where the mean for the subscale was in the moderate to severe higher range. And we came up with 4 items. So one was imitation. A lot of kids are struggling to imitate. This is a very important finding too, because this is a core treatment goal in a lot of autism-specific treatments, is teaching that imitation skill, which is crucial for learning long-term. So that was one to pay attention to. Another is body use. So for this one, we're seeing lots of posturing, repetitive movements, kind of motor things that fall into this category. So that was very common. The third one was this sensory responsivity. So at least moderately preoccupied with sort of sensory input, and it can include either being sensitive or kind of under-sensitive to input. And then fourth, verbal communication. So at this level, in the moderate to severe, speech is often absent, but when present, it can be repetitive, include peculiar, like jargon or pronoun reversal, things like that. The least highly rated item was adaptation to change. So at this lower level, this is below the mildly abnormal range, so kind of within normal limits. Most kids may notice change, but are kind of flexible with it. I didn't bore you guys with all the subskills on the BASC, but there are a lot of adaptive skills, and this actually matched what the BASC showed too. Out of all the adaptive scales, most were in the clinically significant range except for adaptability. So we thought that was also interesting, that we're sort of starting to feel like we're forming a unique profile where we're learning that these kids are socially motivated, potentially more flexible than a typical kid with autism. So to summarize, this data is still very much preliminary, and we're adding people, like I mentioned, every day, but we're excited and reassured that a lot of these measures are kind of jiving together and aligning in what we're seeing. we're seeing a lot of significant symptoms, and that's just made us more excited to keep figuring out the best way to diagnose properly, efficiently, and early, because we're seeing a lot of symptoms, but we're seeing that profile of preserved social motivation and adaptability. So stay tuned, especially for the CARS results. We would caution use of that measure under about 2.5, and we are interested in just seeing how that looks at 3 and above, and if we're seeing even less significance there. And so thank you guys. If anyone has any questions, I'd be happy to answer them. This is our larger team, so all of our collaborators at CHOP and Texas Children's and Cornell, and thank you to the foundation members as well for supporting this work.
Thank you. Thank you. Does anyone have any questions? I see one hand in the back there.
Maybe. Check. Okay. I was just curious, the imitation, did that correlate at all with visual impairment?
We haven't looked at that. That's a good question. I'd be interested to look at that. So thank you for bringing that question up.
We have time for one more question. Here we go. Oh, sorry. Let's do online.
Okay. Sorry, I gotta find it here. Okay. Do you have any age ranges where autism is prolific?
Like most common?
I think that's what the question is.
Yeah. Well, we do think of autism as occurring from birth. It's like a neurodevelopmental condition. So in terms of like the most common diagnosis time, there are, maybe Dr. Reynolds would know this better than me, but I think the average time for diagnosis is like 3 to 6. Does that sound right?
I'm coming.
Yeah.
I think typically it's been about age 4, but part of that is the long wait list to get the diagnosis and that kind of thing. So, and I think it's getting lower all the time with more awareness.
Yeah, great question.
Thank you very much. All right, we are in the home stretch here. We're in our last section for today, and this section we're going to talk about clinical trials. We have 2 speakers, and I'm delighted to introduce the first speaker, who I have not met yet. It's David Campbell from Lundbeck, and I want to. He's going to talk about this in depth, but this is the first trial, I believe, that is actually for across developmental and epileptic encephalopathies, which means that you don't have to have like an LGS diagnosis, and it really opens things up. So for disorders including STXBP1. So I want to welcome David to the stage to give us information on I'm not going to even try. On the longboard trial. Thank you.
Before I jump in, I just want to say, like, the Leadville 100, that is legit. It's not just running 100 miles. Like, you are at elevation, there's climbs, there's descents. Like, that's nuts. Hats off to you. So my name is Dave Chapman. I'm a neuropharmacologist with 20 years of industry experience, and before that, about a decade looking at animal models of epilepsy and seizures. I'm a recent transplant of Colorado to Utah, so when the Michaels moved in, I knew it was my time to go. But I've really been grateful for the opportunity to liaise with some families that are in neighboring states and even in the same county that I'll be in Utah. One fact that is maybe critical for no other time than today, I am the worst dancer on the planet. So I hope to see you out there tonight, and you can try and wear the crown, but I think I've got it. So that's it. But yeah, so what we're here to talk today about is about Bexicaserin and about developmental epileptic encephalopathies. So Bexicaserin, again, it is a treatment that is under investigation. So it is not currently approved for any sort of treatment, but we are looking at it and evaluating it for the treatment of seizures associated with developmental and epileptic encephalopathies, or DEEs. So DEEs as a grouping are over 400 different largely monogenic gene mutations that have a primary common clinical consequence: seizures and developmental plateauing or regression. So STXBP1 is one of these DEEs. Briefly, who we are. Lundbeck is a global biopharmaceutical company who is headquartered in Copenhagen, Denmark. From the United States, we have offices in just outside of Chicago. We're one of the few pharmaceutical companies in the world that is focusing exclusively on brain health. Our legacy in neurological rare diseases started with the first treatment specifically for the treatment of seizures associated with LGS, or Lennox-Gastaut syndrome. That's clobazam or Onfi, and we hope to build upon that legacy. Epilepsy is defined as more than one unprovoked seizure. Seizures are brought about by an imbalance between excitatory and inhibitory neurotransmission in the brain. Two means of restoring this balance in most cases are either to elevate inhibition or to apply some more brakes, or to decrease the excitation, so to lift your foot off of the accelerator. So Bexicaserin is a highly selective molecule that binds exclusively to a receptor called 5-HT2C. 5-HT, 5-hydroxytryptamine, is serotonin. Serotonin is a chemical within the brain that signals across synapses different actions. There are 14 different serotonin receptors, and 5-HT2C is one of them. So when serotonin is released in the brain, it can act on any of these 14 receptors that happen to be in proximity. However, chemists have developed molecules that can selectively bind to some, but not all, of these 5-HT receptors. Within the human body, there are 3 specific flavors of 5-HT2 receptors, 5-HT2A, 2B, and 2C. Bexicaserin was designed exclusively to activate the 5-HT2C receptor. The reason for this focus of specificity is that we hypothesize that activation of 5-HT2C results in anti-seizure efficacy. This is in contrast to 5-HT2A and 5-HT2B, where we believe they don't impact inhibitory neurotransmission, so activation of these receptors produce physiologic effects that are not consequential to anti-seizure activity. So in this case, Bexicaserin and its selectivity is unique. So if we focus on a recently completed study called the Pacific Study, it was evaluating the change in countable motor seizures in patients across a broad group of epilepsies. Patients needed to be on a stable dose of anti-seizure medications, up to 4 of them, and still exhibit at least 4 countable motor seizures per 28 days. Patients were randomized to addition of either Bexicaserin or to placebo, and that is in combination with the medications that they were currently on. So even if they were in the placebo arm, they weren't taking no medication. They were still taking the medications that they were taking before they entered the trial. It was just, were we adding placebo to those medications, or were we adding Bexicaserin? So following titration to a stable dose, patients were maintained on that dose, and then the primary efficacy endpoint was looking at the number of countable motor seizures at the end of maintenance compared to what they were at baseline. From a safety perspective, the most frequent adverse events associated with bexicaserin were somnolence or GI related. Predominantly mild to moderate in intensity, and were most often encountered during the 2-week titration phase. In fact, as it says in that second bullet point, about 1 in 6 patients on bexicaserin discontinued during titration due to an adverse event, and that adverse event was usually somnolence. During the open label extension, when we allowed 3 weeks of titration instead of 2, none of the 9 patients discontinued. In fact, during the open label trial, and I have a poster outside if anybody wants to discuss that a little bit more, only 1 patient has discontinued due to an adverse event, and this has now been going on for over 6 months. So this confirms really a favorable safety and tolerability profile of bexicaserin. From an efficacy standpoint, patients on bexicaserin had a numerically greater decrease in countable motor seizures than did patients on placebo. So you can see at baseline, both of them are up at the top. Bexicaserin, the placebo-controlled portion is the stippled or dotted line. You can see a reduction of almost 60% of countable motor seizures in patients on bexicaserin, and this is in contrast to patients given placebo, which had about a 17% reduction in countable motor seizures. Following that placebo portion, all patients were switched to bexicaserin. And then these are data just looking at the end of 6 months of being on bexicaserin, both patients that were originally on bexicaserin and then those that originally got placebo but then were changed to bexicaserin have a durable response in terms of the reduction in countable motor seizure frequency. So let's move now into kind of where we are currently. We are currently enrolling 2 global phase 3, so these are the largest clinical trials prior to marketing authorization. So this is kind of the go or no-go point in terms of submitting things to the FDA, saying this is why we think this would be important to patients with developmental and epileptic encephalopathies. So we're going to focus on the Deep Ocean trial just because that is something that would be a little bit more germane to this group today. So it is, as was mentioned during the introduction, it is the most inclusive study to date for patients with developmental and epileptic encephalopathy, regardless of what that etiology is. So it includes your favorite STXBP1, but it also includes, again, a number of other conditions that result in intellectual delay or plateauing and seizures. Both of these studies will then go into, so these are placebo-controlled randomized trials, and both of these then will move into an open label extension phase where patients will be able to receive bexicaserin for a longer period of time. Focusing on specific inclusion, again, I'm just going to focus on the Deep Ocean portion. Patients must exhibit seizure types and clinical signs consistent with other developmental and epileptic encephalopathies. Across both studies, developmental plateauing or regression, and must average at least 4 countable motor seizures over a 4-week period for 12 weeks before screening and then during screening. We've already seen a slide very similar to this, but again, just to kind of call out some of the differences. 1, a longer titration period to make that period a little bit more tolerable to patients on bexicaserin. 2, the age of patients is now decreased down to 2 years of age, whereas it was 12 in the study that we had looked at a couple of slides ago. A dramatic increase in the number of patients. So whereas that was around 50 patients total, we're looking now at about 160 in each arm, either bexicaserin or placebo. And then again, that randomization is 1-to-1. So this is my coming soon to a city, state, or country near you. I know that it is monumental to travel. And so if you feel like time or distance will potentially preclude you from making it to one of these sites, let's at least have a conversation. We have resources that hopefully decrease some of those obstacles and barriers. So let's have a discussion if you feel like this might be something that you are interested in pursuing. If I haven't addressed any questions, or if you'd like to revisit this information, the website is shown here. It is the deep-studies.com, or you can email us. Lastly, this is my email, my mobile. If you want to talk to me about, the best bike rides in Colorado or the study, please feel free to do so. But I thank you for your time and attention, and we'll take any questions that you have.
Are there any questions for David?
Thank you. I'm just curious if you have a breakdown of diagnosis of the first. No.
Yes.
Oh, okay.
It's going to be better if I'm talking to a microphone. Yes. And again, we're kind of splitting relatively small patient numbers already, but Dravet syndrome was only 3 patients, and they were all on Bexicaserin. The LGS was in the teens, and then the DEE other was around 20. So we're hoping for a similar equitable split between Lennox-Gastaut patients and patients with kind of all other DEEs. But what we saw in that was, again, and this was kind of what the FDA said, go ahead and try this trial in a broader patient population, was regardless of what that etiology was, the reduction in countable motor seizures was similar.
Awesome. Thank you.
Yep.
Any other questions?
Thank you.
All right, our last presentation of the day. And I wanted to say that all of this work that we're doing to advance progress in STXBP1, it is really based on investing in relationships. And thank you for nodding, Terry Jo, because I really do think it is. And so we have a number of industry partners here, and of course our academic researchers, and these have all, the work that we've been doing has all been investment in, multiple years of working together and building this, hopefully really expansive future for STXers. And I was reflecting that I first met the Capsida team over 5 years ago. So I think 5 years ago and 1 month. And they were landscaping diseases and considering STXBP1. And they just obviously decided to start a program in STXBP1. And here we are now at the start of a clinical trial. And I am, really excited about what Melanie is going to present. And I also feel, I just want to say this as a parent, nothing happens fast enough. Nothing. No progress ever happens fast enough. But going from 0 to clinical trial in 5 years is incredibly fast from a pharma standpoint. And so this is really an exciting moment. And I will pass things over to Melanie.
I thought I'd have to adjust the microphones, but fortunately Charlene got up here before I did it. So I am Melanie Brandabur. I am an adult neurologist. So I spent my clinical world taking care of people with Parkinson's disease. And then about 11 years ago, I moved to ultragenics, where I was working on rare pediatric neurodevelopmental disorders. And so a year ago, plus a couple months, I got a chance to move to Capsida to work on this program. and a Parkinson's program. So it's kind of the best of two worlds for me. And I'm so excited to be here working with this community. So I think there's been a lot of excitement about our program, which I'm very grateful for. And I'm also grateful for Scott and Jim kind of laying the groundwork with some of the not as quite as exciting parts for me, so my work is not quite as onerous as it could have been. So as you know, we are Capsida Biotherapeutics. We are working on a program, an IV gene therapy for patients with STXBP1 developmental encephalopathy with epilepsy. And we are very pleased that the study has now officially started, not with dosing, but with pre-screening of some patients. So very, very exciting. I stole this from the STXBP1 Association website, so it's a very cute drawing. You've already been through some of the importance of the STXBP1 protein. It's really important. In neurology, I always like to say we don't really know what everything does in the brain, but there are some things we've established. For example, that STXBP1 is a protein that's very important for synaptic function. And this is a very cute little drawing of a synapse. A synapse is the part of the neuron where it connects, sends a message to the next neuron so everything can function and messages can get around in the brain. So the synapse is where a little neurotransmitter is released to send a message to the next neuron. So the synapses obviously are critically important because we need all those neurons to talk to each other. So STXBP1, very important for that. Obviously, even kids with STXBP1 have some of that protein, but they generally don't have enough. So we're trying to augment the amount of protein. We already heard a really great explanation of how gene therapy works, so I don't necessarily have to go into that. But the idea is that we have an AAV9 capsid, excuse me, that is going to carry a little piece of DNA into the nucleus, probably will not incorporate into the DNA. As was mentioned earlier this morning, there's a very, very infinitesimal possibility that it could do that, but we don't expect it to. We expect it to just float around in the nucleus and enable the production of DNA, which will then enable the production of the STXBP1 protein. So Capsida is a company that's been working for years on developing capsids made from disabled AAV viruses that not only are safe to put in people, but also will cross the blood-brain barrier and distribute its gene cargo to neurons. That last part sounds pretty simple, but as you heard earlier today, it's not that simple because for very good reasons, the brain is very selective about what it lets in there. If you think about it, you don't necessarily want everything you put in your body to end up in your brain. Think about your college years, right? So it's good that the brain is selective, but if you want a therapeutic agent to get into the brain, you have to always, at some point, wrestle with this issue of how is it going to cross the blood-brain barrier. So these capsids are designed to do that. Typical gene therapy, given intravenously, goes right to the liver, and you always have to contend with that. Not that we're not at all worried about liver toxicity, because of course we're going to be monitoring for it very, very assiduously in this study, but our capsids are designed to avoid the liver, avoid other problem areas, so that more of the actual therapeutic cargo gets into the brain. And that's what we're so excited about, because not only could this work well in STXBP1, could work well in Parkinson's, could potentially work well in a lot of neurologic conditions. So we are very, very excited. But we're at an early step. So in our non-human primate studies, we saw up to 70% of neurons be transduced, meaning be able to make protein in the brain when this cargo was put in there. We saw much less targeting of the liver and the dorsal root ganglia, which are the potential nerve damage that Scott mentioned earlier. And we saw IV delivery which limits certain risks of toxicity. For example, in Parkinson's disease, traditionally, gene therapy has been put directly into the brain, which means drilling a hole in the skull and putting it right into the brain tissue, which obviously carries significant risks. In other therapies, it might be put into the spinal fluid, which is also not without risks. So giving it via an intravenous therapy could potentially, and again, this is an investigational product, but could potentially allow more drug to get into the brain while avoiding the toxicity that's been seen with previous gene therapy studies. So this just sort of shows our expression from several of our capsids across the CNS, the central nervous system. And you can see it's because there is a capillary, a little tiny blood vessel, near every single neuron in your brain. Giving it intravenously allows really, really good access of all of those neurons to the drug. And so you can see we've got really good distribution across not only the cortex, the outside of the brain, but also the internal structures of the brain and cerebellum, the part back here. So you can also see in the diagram on the right that the liver is much less targeted with our therapy. So we're able to give a lower dose, detarget the liver, get more drug into the brain, and we're very excited. Obviously, still very focused on safety, and we'll be monitoring this 24/7. These investigators in the room, I just got their cell phone numbers, so they will be constantly hounded by me through the whole study, and vice versa. We'll be like a syncytium, a connected web of people and information during the whole study. So we announced our IND clearance a few months ago, and I just wanted to talk a little bit about what that means. So in the IND application process, our company gave information to the FDA on our preclinical pharmacology and toxicology studies, the manufacturing data for the drug product, the clinical trial protocol, the investigator information, and a whole lot of other things. I think the application, even though, of course, it was given electronically in this day and age, was about 5,000 pages of information. So after review of the application, the FDA notified us of clearance of the IND. This is not drug approval by any way, stretch, or form. This is authorization for us to take it into the clinic and study it in careful clinical trials. So the primary purpose of this first study, and this is very important, is safety. You've been hearing all day that there are safety concerns with gene therapy. We are very, very aware of it. Our preclinical studies, including our 6-month GPL tox study, looked very good. A little bit of elevation of the liver tests, and then they went back down again, and no other findings. No animals were healthy. They looked fine. But we will still be watching these patients for every minute of the day. So we are now able to go and study this drug in the clinic and see if it's safe. Obviously, we're going to be looking for efficacy, but as we all know, it takes time for things to happen in the brain. It takes time to learn new skills, learn new language, behaviors. As I think Scott mentioned, some of the behaviors could get a little bit worse before they get better. So we're going to be looking at all of that. But the primary purpose of this first study is to demonstrate whether this drug is safe, whether this investigational product is safe. This means baby steps. It means we go very slowly, and we look at the safety data for each patient before dosing the next patient in the early part of the study. This sort of shows, similar to what Scott showed this morning, a rare disease overview. And what I wanted to emphasize on this slide is that, see the red arrow? We are here. So I know some families have expressed a lot of anxiety, like, this is a very small study, what if my child doesn't get into this study? But I want to emphasize that first we have to show that this drug is safe. And if we do, there will likely be more studies and more opportunities. But that's the number one thing, is the safety. So we are here. Other considerations in rare disease drug development include the importance of natural history data. So those of you that are participating in the STAR study, I applaud you, because that data is so important, because at some point, if this drug works, we will have to convince the agencies that they should approve this therapy. And that means demonstrating that what this drug does in the brain is better than a child who's left untreated. And so that's what the natural history study is for, is to establish that baseline, that trajectory. Maybe there is some development over time, which we've seen in some of this data, but we need to show that we are better than that our trajectory looks better. That's why every single bit of this natural history data is absolutely critical. So thanks again for participating in those studies, because they will be very important. The other thing that the natural history study does is shows us which of those many, many forums and tests that you're all doing will help us understand what's happening in a clinical trial. Because as you all know, there are not out-of-the-box assessments that, the box does not say, use these assessments for STXBP1. People like Scott and Ingo and Tuan are developing them in their work, and we will be hopefully helping to validate them in the study, but there are no out-of-the-box assessments. So we borrow from other tools like the Bailey that you heard about today. All of these things are sort of borrowed for rare disease work. So doing a natural history study shows that those assessments are in fact applicable to this population. Very important. The possibility of historical controls. We may be asked to do a placebo-controlled study, but we don't want to, and I don't think you probably want to either. So if there's any possibility that the agency will let us use natural history or controlled data, we are going to try to do that. It'll be a lot of back and forth and a lot of data, but that is one of the things you may be able to do in a rare disease study. There are also potential accelerated approval options. So we have orphan drug designation, and we have fast track designation. So we are hoping that those things which allow better communication with the agency and more meetings and things will speed up the process. So the other thing that's really important here, and this goes back to the natural history study, is that as you can just see watching your kids and everybody else's kids here, they're different. All these kids are different. So it's not like, 200 adult males with hypertension, where you could easily fill a study of 200 adult males with hypertension. These are patients who are different. Everybody has slightly different measures. And so again, the natural history study kind of gives us an idea, like how many of the patients are walking, how many of the patients have 5 words, 10 words, so that we can, again, use that as comparison data in our study. So I think I went through all of those things about rare disease. Right now, we are in the safety part. We're hoping that our phase 2A and hopefully if we're able to expand to a phase 2, that we are able to start seeing some efficacy. And then at some point, we may be able to squish some of these together and possibly do a phase 2 as a pivotal or approvable study. And then there will be some post-approval work as well. So that's kind of the landscape here. Now, the ability to progress along the path to any potential drug approval depends on the data at each step and a lot of other factors and hurdles. So we are here at this red arrow. We hope to progress along this path, but there's unfortunately no guarantee that we'll be able to do that. So that's like my 50 million caveats about the study. We're thrilled, we're excited, we're focused on safety, we're focused on all working as a team, and we hope by maybe next year we'll be able to bring some data back to you. Hopefully it'll be good, and so that's where we are. So let me tell you a little more about the study. It is obviously a gene therapy using an AAV9 virus that's modified so it can't give you whatever disease it was supposed to try to give you, and a transgene cargo. Now, one of the nice things is that this cargo is for STXBP1, the same one that's already there. So since these patients have one gene that's producing STXBP1, we don't have to worry as much about an immune response to the cargo because it's already in there. We have to worry about an immune response to the capsid. And then, as was mentioned earlier, we have to test to see if your child has antibodies already existing against the capsid. And this is going to slow us down because we have to test every single patient in the pre-screening process to see if they have these antibodies. And if they do, unfortunately, they will not be able to enroll in this particular trial. And I think Scott mentioned that we're trying to mitigate that in future studies, but that's down the road. But for right now, patients who test positive for that anti-capsid antibody will not be able to enroll in the study, which is disappointing for all of us. Other than that, the patients must be diagnosed with STXBP1 DEE. Seizures are not required for eligibility. Patients will be taking corticosteroids, dexamethasone and then prednisone for probably about 6 to 8 weeks total. And they'll be monitored as that is tapered. Somebody's happy about corticosteroids. Okay, that makes one person. But that's how we intend to keep the patients safe during the study. And I mentioned the pre-existing antibodies, and there's IV drug infusion, which is the whole point. That's almost the easy part. Okay, this is just sort of a little outline of the seizure, or of the clinical trial design. 3 patients dosed at a lower dose, followed for a month in between to look at their safety data. And then assuming everything looks good after those 6 patients, then we will move to another 6 patients with the highest safe dose. This thing is ticking away here. This just is an overview of the clinical trial with the pre-screening process where you're just tested for the anti-capsid antibodies and your genetic information is updated. Then there will be a screening period, which will sort of be like a waiting room because obviously if we have to dose patients at least 4 weeks apart, there will be some patients sort of waiting to get into that slot. If they wait too long, they'll have to have the antibody test again and could potentially fail out at that point, unfortunately. And then dosing on day 1, follow-up period for 2 years, very important safety follow-up. So don't think you can just pop in with your child, get the gene therapy, and then head to the North Pole, because that's not going to happen. We will hunt you down and do those safety assessments, I'm telling you. So there will also be other endpoints I'll talk about in a sec, but it's all about safety. And then there will be a long-term follow-up study for an additional 3 years. We want 5 years of safety data from this study. It's a big commitment. So the study objectives: safety, 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 safety and tolerability. And then the secondary objectives are all the things that have been being done in the STAR study, plus a few extra ones that I'll talk about. There will be a seizure diary, an electronic seizure diary, whether your child has seizures or not, because in addition to wanting to see if seizures get better, we want to make sure that seizures don't get worse for some reason. Key inclusion, exclusion, there are some other ones, but these are the key ones. Male or female, greater than or equal to 18 months to less than 8 years of age at the time you sign the consent form. Diagnosis of developmental encephalopathy due to an STXBP1 mutation. Stable on medications, including anti-seizure medications. Again, the whole point of a clinical trial is you only want to change one thing. So if you are having to manipulate the seizure meds or some other meds, or suddenly decided your child was going to embark on like a 6-month intensive, like round-the-clock physical therapy program, it's not so great because we just want to have one thing change, and that would be the gene therapy. Everything else we'd like to remain relatively stable. Other investigational drugs within 30 days or fewer than 10 half-lives would be an exclusion criteria. Treatment with an ASO or any other gene therapy or anything like that. Other genes, like if there's a second gene, which there sometimes are, causing similar or overlapping symptoms, we'd prefer to just treat STX1. Those are the main ones. You can look on clinicaltrials.gov to see a more description of all of the inclusion, or most of the inclusion-exclusion criteria. And of course, the main thing with the whole study is you review this with your clinical trial team. They are the medical purveyors of this treatment, and they are the ones who are going to make the clinical decisions. They're going to decide who's eligible. They're going to watch very closely with us for any types of adverse events. You're probably familiar with most of these, partly from today's talks. We have, in addition to a lot of the Bailey assessment tools, we've added a caregiver interview administered by an outside vendor called Lumanity. I've worked with them before. They're very good. The purpose of this is not to make you crazy with yet another survey, I promise. The purpose is to try to see if there's anything we could pick up that our assessment tools could have missed. So they're trained to do this in a very comprehensive and kind way, and they will be doing it at baseline and then at several periods during the study, again, to see if there's any other exciting change, positive or negative, that could have been missed through the trial. There will be a tremor sub-study at CHOP. There will be a QEEG study at all of the sites, looking for changes in the kind of the background rhythm for all of these patients. And I think, and we, as was mentioned earlier, will be using the CSA developed by Scott and his team. We will be using the clinician and the caregiver version of that. Okay, so, I'm down to 0 points here. Okay, or 0 minutes. So, the FDA clearance has paved the way for the trial to go into the U.S., not other countries at this point. We are hoping to open additional countries depending on our interactions with the relative health authorities, because as I'm sure you know, every country has their own version of the FDA. Fun stuff, right? We would like to thank the STXBP1 Foundation for inviting us today, and we are so grateful to the investigators, site personnel, families, and the participants, especially in the STAR study. Thank you so much.
Are there any questions? Oh, here, we'll start here. Go ahead.
I was just curious about the antibodies that you were talking about of the virus. So is that, you didn't really specify exactly what the virus is, but
yeah, I can talk about the antibodies. So basically, all of us get exposed to viruses all the time, right? And AAV viruses, every kind of virus. And so our body makes antibodies against those viruses, so that if anything that even looks remotely similar comes along, your immune system will attack it, so they don't kill you or whatever. So you have all of those antibodies hanging around in your body, and if some of them look similar to our capsid, they will jump on that capsid, disable it, and potentially make your child sick. So we have a special company that's made a test to detect antibodies that are to our capsid, in case anyone's picked those up along the way. We don't know how many patients that will eliminate. We will be gathering that data as we go forward with the pre-screening. In other studies that have looked at a different capsid detecting test, it's been, for the pediatric population, it's been around 20% of patients. So we're hoping it's less, could be more.
Julianne, or?
That was my question. Okay. I have a question about any potential publication or early sharing of findings as you go through the phases until the pivotal phase. Is there a schedule or anything to understand what's happening?
The question is about early sharing of data. So I don't think, this is obviously above my pay grade to decide when we share data exactly, but I will say that we're obviously going to be very careful. We're not going to be like, oh, we dosed a patient, oh, we dosed another. We're going to, because we need to look at the safety data, decide if we dose the next patient. So I will say that we will, I'm hoping by this time next year, we'll hopefully be back here to share some data. And between that, there may be other data released. I will tell you that Charlene has me on speed dial, so if we have any data to share, she and her team will drag it out of us and put it on her website, right? So.
And you may want to mention that you already have like a resources area for the preclinical data that you have on your website?
Oh, yes. There is a lot of information on our preclinical program, presentations to like the ASGCT meeting just a few months ago with our preclinical safety data. And yeah, there's a lot of information on our website. I think it's kind of hard to find, but I think if you go under news, you should be able to find it under news.
Any other?
I have an online question. Someone would like to know what is the, I apologize, what is the actual virus you're using for the capsid? Is that possible to talk
It's
about?
an AAV9 virus, but beyond that, I think it's a state secret and I can't tell. Sorry.
Any other questions? I have a question. So in your, kind of your broken out phases or stages of the phase 1/2A trial, there's a 2-year long-term follow-up. Does that mean that in order to go to the next phase of the trial, you're going to wait those 2 years?
No.
And because I think that this has also been brought up because there's been a question around.
That's a great question. So the early study will follow patients for 2 years, and then there's a 3-year add-on study. But during that time, as we get data, we will have additional talks with the FDA and the other health authorities in other countries about what the next part of the trial will look like, what assessment tools we should be focusing on, what additional safety data they might want. So that will be ongoing. It's not like we're going to say, 5 years, okay, now we can look at the data. It doesn't work like that at all. But thank you for bringing that up. That is a question we have had before, yeah.
Any other questions?
Hi, I know you said that you didn't know whether or not the FDA would require a placebo for the clinical trial, but when that placebo, if it's necessary and it's delivered, would it be delivered via the same virus vector, or would it just be something that wouldn't create any antibodies in the children?
That's a great question. So I just want to make clear, for the first study, no placebo. It's open label. Everybody who participates in the study will get the treatment. There's no placebo in this. I'm talking about down the road. It's possible that we'll have that conversation. And then as to how that happens, it's a really interesting scientific question because, like, for example, in other studies they've done, in Parkinson's studies, they used to do a burr hole in the skull so that you couldn't tell, if you were sitting next to somebody in the waiting room, you couldn't tell, they didn't really drill a hole in your skull, so you must have gotten the placebo, So there are a lot of different ways to do it. You can do it with a sham procedure where you get an IV with just saline in it. Don't know. There are a lot of interesting ways to do it, but it's a very good question. And I don't quote me on this, but I think it's unlikely that there would be an empty capsid given. So, great question, though. Any other questions?
One more.
Okay. How will y'all decide how long to follow these children around? Like, is, age expectancy, like, if the trial continues to have positive effects, or, like, when are you going to do the cutoff? Like, you've received the IV, so, like, we're going to follow you for 5 years, 10 years, like.
So 5 years is kind of the standard time for following a patient after gene therapy at this point in time. Obviously, that could change. It's different in other countries. I think in the UK, it's 15 years, for example. So that's kind of like the standard. In most clinical trials, you wouldn't follow someone for that long, but in something like gene therapy that could have longer-term ramifications, 5 is kind of the current standard. So part of it is when we expect to see safety signals, but also what the agency's decree is the right length of time, and it could change. Other questions?
Do you expect the age range to change, like, as things progress?
That is a very good question. Do I expect the age range to change? I don't know, because I hope it changes. I hope we're able to address both younger and older patients at some point. It'll depend on our safety data and our efficacy data. As Scott mentioned this morning, two schools of thought on things like developmental windows, like when you could make a significant difference by giving somebody a therapy. And there are a lot of things involved, like what the disease is, what the mechanism of action of the disease is, what the therapy is. But there has been data and some other studies for other conditions that maybe you can see improvements in a teenage population or above. So I'm hoping that once we have the data for the younger population, we could expand it in both directions. But we don't know yet. We just don't know.
Yeah, it was a great talk. Thanks so much. And I was going to actually ask about the age as well, because in the adult mice, it looks like they see reversal of a lot of the symptoms even at that age. But I was going to maybe ask also about the first dose, and if you were able to agree with the agency on a dose where you might expect to see some benefit.
So I will say that in terms of, this is about dose and benefits. So in our clinical trials, you heard from Min-Shan earlier that there does seem to be good treatment effects in the mouse model, and then so we use that to sort of establish what might be a therapeutic range. And both of the doses we're taking into the clinic are in that range. So we expect both of these doses to be therapeutic, as much as we can tell that with the data we already have, since we haven't given this to humans yet. So we're optimistic that both doses should be potentially therapeutic. Does that answer your question? Okay.
Any other questions? I have one question. What was the age of the primates who were part of the tox study?
The age of the primates, I don't know that off the top of my head. I can get back to you with that. It doesn't exactly correspond well to human ages, so it's different, but I don't know the exact ages of the primates. I don't know if there's anyone here who does. Is Swati here? Oh, there you are.
They're not adult primates, so I can't give you I am loud, but
Yeah, these were juvenile primates, so not adults. So they were young, on the younger range, similar to what we're looking at for kids. But like Melanie said, it's not a one-to-one correspondence.
Thank you.
Jared.
Thank you. I've got a question from online that's asking, can we get more information on the dosing? Will the first 6 patients get the higher dose, or will the first 6 get a low dose? And if all goes well, then the next 6 get a higher dose?
So the way the study is structured right now, the first 3 patients will get the lower dose with minute examination of all of the safety data, and then assuming it all looks good, we will go up to the higher dose for the next group of 3 patients. So that's the phase 1 part of the study. And then we will have a committee of experts meet and review all of the data. And if everything looks good and we aren't seeing a safety signal, we will most likely move to the phase 2A part, the next 6 patients with that higher dose. Sorry if I didn't make that clear.
Any other questions? All right. Thank you very much.
My pleasure.
All right. Well, we have definitely had a full day. It was so much information today. My head is full. I'm sure your heads are full. But hopefully it has really given you an update on what is happening from a research perspective, and what we're really seeing in the STAR study and other studies. So you have a break now. Oh wait, Melissa is going to announce things, not me.
All right. Okay, so I know your brains are full of information. On your tables, remember Amanda's offering that session. It's just kind of a processing session, seriously, just to talk with some of your fellow parents, to kind of talk through some of these things if you're interested. I know it's warm outside, but it is going to take place outside in the courtyard. So find some shade. Amanda will meet us out there, and we can do that. In addition, while you're leaving, our photographer has arrived. If you would like to take a quick picture with your family, she will also be here this evening, though. Okay? So don't feel like you need to rush, but she is going to be out by the STXBP1 banner. She's waving right here. She's right here. She's going to come see you. If you want your picture taken now, she'll do it. Otherwise, you can do it this evening at the dance party. Don't forget about the dance party. Okay, we'll serve you some food, too. We'll serve you some food. Oh, when should you come? You should come at 6 o'clock. Dinner will be served at 6 o'clock, and then we'll get going with some fun awards, and then we'll have our dance party. All right? So we'll see you guys back here at 6 o'clock.